Welcome to the Global Health Crisis Reporting Forum and our series of webinars on the COVID-19 pandemic. And my name is Patrick Butler. I'm the Vice President of Content and Community at the International Center for Journalists, which sponsors the Global Health Crisis Reporting Forum. And we have a very important webinar today on vaccines and some of the misconceptions that are traveling around about vaccines. They're finally here. They've arrived on the scene with an explosion of information, some of it good, some of it bad, and some of it downright dangerous. Joining us from Harvard Medical School is Dr. Galit Alter, a professor of medicine and a group leader at the Ragon Institute of MGH, MIT, and Harvard. And we're going to help journalists and their audiences make sense of the infodemic on vaccines and the science behind them. Dr. Alter's work focuses on the development of systems biology tools to define the correlates of immunity against infectious diseases that ravage the globe. Through this work, she seeks to advance rational vaccine design. Dr. Alter is a two-time recipient of the prestigious MGH Research Scholars Award, and she was elected a member of the American Association of Microbiology in 2019. Welcome, Dr. Alter.
Thanks, Patrick. So I thought I would put some slides together for this particular presentation to give you all a bit of an overview of how we think about vaccine basics, if that's okay.
Great. Before you do that, I just want to do a couple of housekeeping things on reminding people about how to ask questions and that kind of thing, and then we'll share your presentation. So just a couple of reminders. If you're on Facebook, search for the ICFJ Global Health Crisis Reporting Forum group and join it there. We are running forums in 5 languages: Arabic, English, French, Portuguese, and Spanish, and more than 10,000 journalists around the world are participating in the forums. If you're not on Facebook, you can go to the COVID-19 resource page on ICFJ.org, where you can sign up for newsletters to get news on upcoming webinars. You should also visit ICFJ's publication, ijnet.org, which is the go-to place for stories on the latest journalism trends, tip sheets on just about every practical aspect of journalism, and opportunities for training and career advancement. That's all in 8. Please type your questions during the discussion in the Zoom chat on the icon below. And if you're watching on Facebook, you can post your questions below the video post, and my colleagues will forward them to me. So we're going to welcome your questions a little bit later in the program. So now Dr. Alter is, as she said, going to give us a presentation that will help us understand the science behind the vaccines. Thank you.
Super, Patrick. Okay, so I am going to just walk you guys through some of the basics. We call this Vaccines for COVID 101. So the idea for us was that back in January, a decision had to be made in the vaccine community to try to decide how to engineer vaccines as fast as possible. This is really in the wake of just receiving information about the sequence of the actual virus. Now, while some vaccine developers decided to use the entire virus as a potential platform to drive immunity, really following on the heels of some of our first vaccine inventors like Louis Pasteur, most vaccine developers actually ended up focusing in on a specific part of the virus, which is the spike of the virus. So what I'm showing you over here is what the viral particle would look like in a cartoon form, where the protein that is most abundant on the surface of the virus, the protein that's involved in infection, is really the spike protein that, as I'll show you in just a few minutes, is really the dominant target for most of our immune responses. Okay, so the way that the virus causes infection or causes COVID-19 disease is really due to its ability to use the spike protein to interact with a protein on the surface of our lung cells, a protein called the ACE2 receptor. This is the dominant receptor for the virus. And once that spike protein can interact with the ACE2, this gives permission essentially to the virus to now enter the cells where it can replicate, multiply, and begin to spread and cause disease. So this interaction is really the most important interaction that allows this virus to infect. And this is the interaction that we seek to block by using vaccines. Okay, but what's interesting about this particular ACE2 receptor is that it is not only present on the surface of lung cells. These are these cells here that are found in the upper respiratory tract or in the lung. But this receptor is also found in the gut. It's found on our blood vessel walls. It's found in heart cell wall, as well as found in some cells that live in, for example, our respiratory passageways where we can, allows us to smell and taste things. So the virus is actually able to infect many other types of cells in our body, which is often why in the media we hear about reports of off-target effects, so non-lung associated different kinds of symptoms like diarrhea, some sorts of other types of vascular disease, as well as downstream sometimes renal disease and cardiac outcomes. So this
And
is
is that also why people can't sometimes smell or taste?
correct. Very good. That's right. So that's why one of the most common symptoms and one of the most unique symptoms of COVID-19 is the fact that we lose sense of smell because it infects our olfactory neurons that are the neurons that basically sense smell and taste. So really, really fascinating. Okay, but these are mostly off-target effects, and the virus tends to replicate in these particular cells, and this is why typically the disease is contained within the lung. But so what the body does to fight against this virus is that the body begins to make antibodies. So what happens in the fight against the pathogen is that there are these magical cells called B cells that are my favorite cells that are essentially our antibody producing factories. They recognize the virus, they begin to make antibodies, they mature them, they make them really into these magical bullets that these B cells can then secrete and make in humongous numbers. Imagine one cell can make 2,000 antibodies per second once they develop. And so these types of cells start to spew out billions, if not trillions of gagillions of antibodies that are essentially poised to essentially bind the virus. And if they bind the virus in just the right way, these antibodies can then block infection, really interrupting that step of the virus hitting the cell, so essentially ablating spread and potentially limiting disease. Okay, so following infection, what happens is that the body starts to make antibodies to the virus. Once the virus is cleared from the system, these antibodies persist, theoretically giving us long-lived immunity against the actual virus. Probably as you've heard in the media, there's been this almost schizophrenic, are there enough antibodies? Are there not enough antibodies? Are they persisting? Are they providing protection? The answer is that we don't know what the precise level is of antibodies that is needed to confer protection. But the one thing I can tell you is all the vaccines that are currently being tested in humans or already licensed induce such high levels of these antibodies that it does not look like a virus can really cause too much damage if full vaccination is achieved. Okay, so as I mentioned before, in the vaccine development community, we had to make a decision very early on. And what the vaccine developers that were not using the full virus decided was to essentially focus in on this one particular protein, the spike protein, which is essentially the target we believe is the most important for antibodies to provide protection from infection. So most of the vaccines, are really based on designing ways of exposing the immune system to this particular part of the virus, the part that is most important to hit with an immune response. So there are multiple different platforms out there for trying to drive immunity to the spike protein. As many of you have heard, first in line that is really, already licensed and already out there in many countries are the mRNA vaccines. So these I'll tell you about in a minute how they work. These are really fast, very flexible, and that's what makes them very attractive as a new vaccine platform because you can essentially make this vaccine in a really short amount of time. The challenge now is scaling it up to get enough doses for the entire world. But the cool thing is that you can redesign it very quickly, which is becoming potentially an important aspect of vaccine development, given the emergence of new viral variants of concern. Do you have a question, Patrick?
As I understand it, the mRNA vaccines have never been approved for use in humans until these vaccines.
That's right. It's our first time using mRNA vaccines. It doesn't mean that we haven't been working on developing them for dozens of years. It is just that this is the first time they're being approved for licensure for large scale deployment. So it's really important to know that mRNA has been in humans for many, many years. We've been using them for different kinds of therapeutic indications. This is the first time that we're deploying them at a global level to fight a single viral infection. Okay, we also had DNA vaccines. This is another form of genetic vaccine that was being developed much earlier on. Back in the 80s, we were trying to make DNA vaccines for many different diseases. They're harder to make, they're harder to use. If you're interested in trying to understand why they don't do as well as mRNA, we can discuss that separately. But there are some efforts in trying to develop DNA vaccines as well. Okay, second in the queue are the inactivated viral vector vaccines. These are the adenoviral vaccines that you've heard about from Sputnik, from Sinovac, from J&J. These are all essentially, they use the outside of a common virus that it basically circulates in humans. It doesn't cause infection, it doesn't cause disease. These are essentially like innocuous viruses that circulate in the population. And what makes them so awesome as vaccine delivery vehicles is they kind of act like a little Trojan horse. They go in and out of our cells all the time. They go in and out of our bodies all the time. And what we can do is we can essentially spike in a little bit of the coronavirus protein, that spike protein. And now instead of just delivering their own nuclear material, they're also exposing the body to the coronavirus spike antigen, which allows us to now make antibodies to this target in a completely natural way. It's super, easy to imagine how this works. And the nice thing about these particular vehicles for making vaccines is that they can be produced at huge scales, which makes them advantageous over the mRNAs. They're slower to design, but way easier to scale up at a global level. And they've been in thousands of individuals. And so we know these kinds of vehicles are very safe. So that is a second platform that we've been using. Okay, the third platform dates back to some of our earliest vaccine development approaches that date back to the 1900s, where we had essentially come up with a strategy that you can actually just make proteins or isolate proteins from a pathogen and use them to basically expose a system to that foreign material so they can make antibodies. So these are essentially just pieces of proteins that we make in giant vats that we then purify using these standardized, really well-established methods. And then we basically package them up in a vaccine kind of solution, and then we can administer this to the immune system to drive immunity directly to the proteins. The problem with this platform is that it's a little bit slower than these other platforms, right? Because you've got to do all of this quality control to make sure that the proteins are in perfect state. But this is one of the most traditional approaches that we have been using for, 100 years, especially for vaccines like flu, which is based on this same technology. Okay, and finally, the last platform that is being used predominantly in the East, or I should say in Asia, is an inactivated SARS-CoV-2 particle, where we can essentially take the entire virus and using different kinds of treatments, we can essentially kill it. So it is still present, it is still intact, but it cannot cause infection. By using this type of method, you can essentially expose the immune system to the entire virus in its intact form, but that virus is crippled and cannot cause disease. The advantage there with this platform is that this exposes the system to all the different parts of the virus, not just the spike, which might give us advantages in the long run if we believe that other targets of the virus might also help to band-aid or remedy potential breakthroughs in new evolving viruses. The problem with this platform, I just want to mention, is that it is a little bit slower also to produce, because you have to make huge amounts of virus and make sure that every single virus in that vat that you've essentially brewed is completely inert. So there are really important chemical processes involved in basically making sure that the product is safe, but that is something that just takes an extra bit of time and really is a concern for most regulators. But this is one of our oldest, most traditional approaches, also invented by Louis Pasteur, as I mentioned before. This was invented in the 19th century, a really important way that we've made vaccines for hundreds of years. Okay, so how do these platforms work? Okay, so here's an mRNA vaccine. Essentially what it is this is just the genetic code for making the spike protein. There's nothing else to this little string of nuclear material that literally is like software that we use to basically teach the system how to make the protein without having to make it ourselves in a giant vat. So we're letting the immune system, our body, to make the protein itself. The way that we deliver it is essentially in fat particles. So these are fat particles that we have in our own system. These fat particles essentially coat all of our cells. And the reason we do this is these fat particles, they coat and they protect the mRNA. So when it comes in contact with a cell, let's call this the cell, this big yellow blob. When it comes in contact with this, the fat particles from the mRNA vaccine fuse with the fat particles on the surface of the cell. It essentially allows the mRNA to essentially blend right into the cell membrane. So it doesn't affect our cell, it doesn't hurt the cell at all. And essentially it allows the mRNA to essentially get right into the cell. Once the mRNA is in there, it doesn't even go close to our own nucleus or our own DNA. It stays completely sequestered and locked away. And all it does is it basically provides the software code for how to make these spike protein. So our own cells machinery recognizes these mRNA strings using totally natural processes that we use typically to make proteins that build our own cells or build our own bodies. The mRNA is essentially translated and it leads to the generation of proteins that are then released outside the cell. They get picked up by cells of the immune system that then tell those magical B cells or antibody producing machines to make lots of antibodies to fight the disease. This process is totally natural. There is no way these mRNAs can alter the actual host cell. And all it does, these mRNA vaccines, is essentially delivers one extra piece of software that the cell then essentially helps it make these proteins that can then educate the immune system. Okay, so the viral vectors do something very similar, but instead of just blending into the surface of the cell, what these viral vectors, these adenoviral vectors do is they have these special receptors on their surface that give them access to immune cells. This allows them to attach to different kinds of cells in the system. It allows them to dump their own viral cargo into the cell. And in the exact same way as the mRNA, these codes delivered by the vaccine are then essentially translated into proteins that encode the spike protein from SARS-CoV-2 that is then produced, picked up by immune cells, and then delivered to B cells to make these magic bullets we call antibodies. So the process is super simple. The one difference is with the protein-based vaccines. So with the protein-based vaccines, these are the proteins here. For many of the developers that are using this right now, they have done a little bit of a tweak on the original design strategy that was used historically for flu and for other vaccines. Instead of just giving protein alone, what they have done now in the context of the COVID-19 pandemic is they've added new immune stimulants to the vaccine. These things are called adjuvants. Adjuvants are essentially innocuous molecules. Again, they don't cause any disease or any kinds of strange manifestations. But what they do is they really get the immune cells activated. They're like little kind of like, I would call them like, they're like candy for the immune system. They stimulate the immune system and they help them recognize these proteins more effectively and help them make really large amounts of antibodies that we need to essentially arm ourselves to fight the virus wherever we are and however we are exposed. So this makes the protein strategies a little bit different than the historical protein-based vaccines, but essentially this is the mechanism where instead of having to make our own cells make the protein, here we deliver the protein already to the system, allowing the immune cells to pick it up and then to make these wonderful antibodies that protect us. Now, one of the big questions that's come up in the vaccine, in the media, I should say, is the question of how do vaccines work, right? So we're talking about this kind of, I think, this like mess of different kinds of clinical outcomes. So some vaccines will work by completely blocking infection, right? So what they will do is they will make sure that nobody becomes infected. So everyone will stay essentially dark. In some instances, what we're asking is the vaccine to basically just limit disease, right? We want to keep people out of hospitals. We want to keep people alive. And so what we're talking about is trying to keep people away from this hot, red, infectious state, right? But if we have a couple people who have a cold, maybe that's okay, right? Because if we could turn COVID-19 into a cold for the whole world, I think we could live with that, right? That would get us out of masks. That would get us back into our, barbecues with our friends and back in churches and synagogues and mosques and everywhere else you want to go. So the idea really is that if you have a population where you have one person who's severely ill, one person who might have this cold-like symptomatology, the question is, what do the vaccines do? Now, if you have no vaccine, what we know is that this particular virus will spread very quickly. And unfortunately, in a completely unpredictable way, we don't know who's going to get mild or severe disease. And that's scary. And that's why we stay in quarantine. This is why we are still confined to the social distancing measures that we have across the world. One possibility is if the vaccines are good and they can block disease, then we can go on with our lives. It's okay. A couple people might get colds. It's going to suck. We're going to need Kleenex and maybe some extra chicken soup, but it's not going to stop us from living our lives. And this is really an unbelievable opportunity to just get to this level. But what people keep wanting is this idea that we could have a vaccine that can completely block transmission, meaning that we're not going to have any colds and no severe disease. Now, this is obviously the icing on the cake. This is the absolute dream of what we could get. And this is what data out of Israel is kind of pointing to, that at peak, immunity, when we just after vaccination, that we might be able to really begin to limit the disease. But what I would like to advocate here is that a more reasonable option of what we should be shooting for is transforming COVID into a cold. This is a realistic dream. We want to get away from this. I don't think we need this. If we get there, awesome. But I don't think that that is necessary to get us out of the current pandemic. Okay.
In that middle option, just go back one sec. In that middle option, where it looks like we have a few people that have the cold-like symptoms, we can expect that there will not be people who get the severe need to be hospitalized.
That's what the data suggests, right? So that's important, Patrick, because what we're reading out in all the phase 3 trials that have been done so far all across the globe, what we're reading out is the level of severe disease or hospitalization. So we're not reading out in any of these trials transmission blocking. We are only looking at the level of efficacy of these vaccines for limiting the red. That's what we're trying to limit. And that's a really important distinction, because it does not seem, I mean, at least, from a cost of trial perspective, trying to attain this was really going to be difficult. Okay, it was going to cost us, trillions of dollars to do these kinds of trials. Mm-hmm. And on top of which, it's not clear this is a reasonable expectation from a respiratory pathogen. It might not be reason. It might happen, but that is not what we were reading out in these trials. And so what we've been reading out is this phenomenon, right? Is we're trying to look at, and it's important to then divide up the data, because everyone's reporting it slightly differently. Some people are reporting out, this is how many people die, this is how many people have hospitalization, this is how many people have severe disease, this is how many people have mild to moderate disease. Mild to moderate disease, of course, we'd like to eliminate it, but it's not that big a deal, right?
It's the common cold is a coronavirus, right?
It is a coronavirus, and there's other viruses that cause common cold, but yeah, common coronavirus can cause a common cold. The problem is that in order to get to this, right, we're going to have to get a very large fraction of the population to get immunized in order to make sure that we're not going to make a few people who get the vaccine last, right, get severe disease. Because even when you're in this state here where you have a common cold, that means these guys are carriers. And so everyone's not immunized, right? Then you can still be a spreader. So this is why we have to continue to wear masks until a sufficient number of our population is fully immunized in order to prevent the possibility of carrying the virus, right, as a common cold carrier and giving it to someone who has not been vaccinated yet. So there is still some nuances to how we have to think about this, because if this was the outcome on the right side, then it's essentially we could all take off our masks because there's no chance of getting infected, but that is not what we're reading out. And that's why it's so important as a media group to be able to communicate this information to the world. This is what we're reading out. This is what we're measuring. And this is why we have to continue to wear masks until we can protect everyone who's vulnerable to the disease. Okay, so what are the data? So I'm going to just show you the data that's been published so far in scientific papers. This is not to say that there's not other vaccines that are being tested globally, but these are the ones that have been published in, highly vetted, scientifically, rigorous journals. So Pfizer with BioNTech, Moderna, and Sputnik have all reported 95% reduction in disease. That's severe disease. So not reporting blocking transmission, they're essentially reporting whether or not they're able to keep people out of hospitals and, alive. They're also reporting somewhere between 30 and 50% reduction in transmission. And that's a very interesting figure, because if that is true, then that means that we might get to that second ultimate kind of outcome, but that's not a guarantee. And so I think we have to continue to look at the data to be really sure that it's safe to take off our masks. And 30 to 50% is probably not enough to completely protect everyone from being a super spreader. So we have to be careful. Now, the other thing I want to mention about this is that for all of this data, and this is one of the most important things I can communicate to all of you today, is that we're reading out vaccine efficacy at the height of our immune responses. Just after we've been vaccinated, our body is so full of these amazing magical antibodies that it's no wonder we have this wonderful outcome in this response to the virus. I don't think as vaccine developers, we would have imagined we would have hit these numbers, but whatever, we're super happy and we cannot believe how effective these vaccines are. But the point is that these levels of antibodies are going to go down over time. This is the nature of immunity. Right? We have to make space for other potential pathogens in the future in our immune systems. So the question is going to be, how long are these levels going to last? And so that's why it's so important to take this with a grain of salt, is that we're hitting 95 and 30 to 50 at the peak of our immune response. We have to keep watching for the next 6 months, 1 year, and 2 years to see really where is that sweet spot going to live once we get down to a steady state of kind of sort of population level immune response. Okay, AstraZeneca.
That leads to the question.
Go ahead.
No, I was just going to say that leads to the question of whether we may need to get revaccinated frequently, and I know you're going to address that.
Correct.
In a bit.
That's right. So AstraZeneca is a little bit trickier because the vaccine data has been a little bit more variable, but depending on where they've tested and what regimens they've used, but they're hitting somewhere between 60 and 90% reduction in severity of disease. And they're reporting about 60% reduction in transmission. Again, this is always at peak immunity. Johnson & Johnson, interestingly, is showing 85% reduction in severe disease, really, and death, which is so important with a single shot. It is the only one with a single shot that can hit these levels, right? And that's really important from a global deployment perspective. Having a single shot to get you to that level of immunity is amazing, right? It is just outstanding. Yeah. And so that is a pretty outstanding result at 85%. And then Novavax.
Is that one of the ones that does not need to be kept super cold? Or I know some, yes, okay.
So this is the one that can sit at room temperature for a long period of time. You can make tons and tons of it. It can be deployed globally pretty effortlessly. And already Johnson & Johnson has used the same platform to vaccinate much of the DRC already with Ebola vaccine that has been really remarkably effective as well. Okay, so Novavax is the last one that's coming up on the radar in the next few weeks. This is a protein-based vaccine, and this one has shown 80% reduction in disease in the United Kingdom, where there was a mutant that was circulating a variant, and 50% in South Africa, which is really kind of interesting. So that means that you still get efficacy, although not as high against some of these new variants that are emerging. And I'll talk about that in just a minute. But this is pretty still outstanding because that means that we have a huge toolkit of vaccines that are at our disposal. We have never in history had this many vaccines to go against a single pathogen all at once. So this is really a momentous time in vaccine development. Okay, so what about the variants of concern? This is an issue that I just really want to raise with all of you because I think this is another point in the media that's kind of driving me a little bit nuts. So we have these new variants that are popping up in the virus. All viruses will evolve. It is a complete misconception to think that this is unusual or unexpected. We were all waiting for this to happen because this is the nature of viruses, right? They have to adapt to their hosts. The virus transited from a bat into a pangolin and then into a human. And so of course it has to adapt to its host as it essentially evolves in this new species. So these mutations largely affect the ability of the virus to interact with its receptor ACE2. But once in a while, one of these mutations can occur at a site on the spike where it can block antibody binding. And that's where we get a little worried. So the reason we get worried is that if there's enough of these mutations that appear, some of these mutations can eventually block antibodies that are present following vaccination. So the question is whether or not when these new variants emerge, are they essentially blocking all of our antibodies from functioning, or are they only potentially disrupting a small number of the antibodies in the system? Because if it's only a small number, then that means we can still get good levels of protection. It's just that maybe not all the antibodies we generated are going to be perfectly adapted to fighting the virus. So what it turns out is that from the data we've accumulated from many, many groups globally now, is that we see that there are many variants of concern that are essentially emerging across different parts of the world. They are not emerging solely in the UK or only Brazil or only in South Africa. The same mutations are popping up everywhere. There is the same adaptation to all humans. And that is something that we need to basically help the population of the world understand, is that these are not only coming from specific populations, but they are likely to emerge in every city, in every village, in every country all around the world. And the same solutions are being used by the viruses, which is just amazing from a nerdy biologist perspective. Okay, so the point is though, is that some of these antibodies that we make with vaccines are still binding, right? The Pfizer-BioNTech vaccine still binds, Moderna still binds, and we still see efficacy with J&J, and we still see efficacy with Novavax. What the problem is, and this is what we're seeing in many of the data that are being published, is that this is the level of antibody blocking activity on the y-axis that we had against the original strain. Now with the strain that was first identified in the UK, we get a slight dip in the ability of antibodies raised by vaccines to block infection. And with the strain that was first identified in South Africa, again, we get an even bigger dip. But the point is that we still might be able to reach herd immunity. If the antibodies can provide sufficient protection that they can keep people out of hospitals and keep them alive, then that still might be enough. Even more importantly, if you look at protection from death and severe disease in the US, UK, or in South Africa, where many of these vaccines are being tested, we're still getting good efficacy, which means even if we're not at 95, even if we get to like 60 or 70%, this still might get us to herd immunity. So what is herd immunity? This is the most important point, hopefully, that I can make. And I'm sorry, I know I've gone over, so maybe I'll stop after this.
No. Please go ahead.
Okay.
This is really...
Okay, so if you have no vaccine and you have one infected person, this is a population of people that have not had the vaccine, right? This person very quickly will start to spread, right? And cause more people to turn red. And very quickly, you see the whole population will get infected, right? This is really important, right? This one person might make antibodies spontaneously, and maybe that protects him from getting reinfected. But overall, the virus just spreads throughout the entire herd of humans. If you have a super protective vaccine and you turn everyone into these little green people, not Martians, but immune people, then essentially what happens is that even if you have an infected person, that virus can't get far, because at a population level, you have stood up immunity so robustly that the virus cannot get around all that immunity in the population. And that is where the herd, or the group of individuals, is more valuable than the one person who might have been vaccinated, right? We're standing up immunity around people who might be more vulnerable. So these people who may not have gotten access to the vaccine, for example. And you can see that there's a limited spread of the virus. Now, what happens if you have lower protective vaccines? So even if there's a viral variant of concern, right, and you still get a sufficient number of the population immunized, then maybe you're still going to have a few more vulnerable people in your midst. But the important part is that with enough green, you're going to keep that virus away from most of the vulnerable people. So what that means is that even if your vaccine is not at 95%, we can still hit herd immunity if we vaccinate a sufficiently large fraction of our population. Okay, so let me do
How
this
do
one.
we, maybe you're getting there, but how do we determine what that level is?
Oh,
We're all
yeah.
worried about people who refuse to take the vaccine, and you know, the percentage of those will make an effort on whether will have an impact on whether we get to herd immunity, I think, right?
Yeah, it's a really important question, Patrick. You know, so the epidemiologists have been working very hard to calculate that number. At the beginning of the pandemic, I think we thought it was around 60% of the population had to get immunized in order to get protection. I think we've realized that's not the case. We're going to have to get closer to 75% of the population. You know, education is the only way that I think we can get around, you know, the worries that people have about getting vaccinated. And really, I think that what it is that, you know, I think there's two kinds of anti-vaxxers. I think I don't want to call them anti-vaxxers, but people who are vaccine hesitant in the world. There are people who are just really against vaccines completely because they're just not fully aware of the global impact that vaccines have. And they've heard anecdotal stories about potential side effects, which are completely have been debunked and proven to be completely untrue. And then there are people who just don't know enough, right? And those are people who, with enough communication and enough education, and with, you know, people who are able to answer their questions, I think we're going to be able to turn it around with these individuals. They just want to understand what they're putting in their bodies. And that's, I think, what we're here for, to help folks understand that these products are so safe. These products are simply just a way that we just teach our immune systems to fight a pathogen that can do much, much, much more damage than, you know, the vaccine itself. It does almost nothing to the system. It doesn't alter any kind of immune response. It doesn't change your life expectancy. It doesn't change anything in your system. It cannot affect your brain. I mean, it's just amazing, the funny anecdotes that get picked up by people that, you know, we as scientists have investigated so deeply and proven to be completely false statements. And
this is why you're here talking to us, right? Because the media is so important in getting that message out.
that is absolutely right, right? You are our voice. You have to tell us what information we have to give you so you can help people understand the value of vaccines and really, you know, just how marvelous they are from a scientific perspective and how they can just train the system to do something that it should do on its own. We just help it do it a little faster and a little bit better. Okay, so let me just give you one more little tidbit before I go to the safety data. And I just want to say that, you know, one question that keeps coming up in the media is this going to be a once in a life vaccine, or is this going to be a yearly vaccine, right? So we don't know. But for flu vaccine, what I'm showing you here on the left side is the fact that, you know, for flu vaccination, we have to change the vaccine every year because the virus changes every year. The reason that flu changes so rapidly, and these are just some of the bigger changes that have occurred in flu, is that flu, its genome is actually composed of 7 segments. And when a virus, one flu virus, comes in contact with a second flu virus, it can essentially reassort these segments of its DNA, or its RNA, actually, I should say. So what it does is it reassorts really efficiently. So flu can change in ways that coronavirus cannot, at least that we know of yet. It can essentially reassort itself every year and create brand new variants of the virus that we've not seen. And so we're lucky that so far flu since 1918 has not really reassorted to something really pathological. But this is why so much of our effort in the vaccine development community, it has been focused historically on trying to develop universal vaccines against flu, because flu is just a complete biological marvel in how it's evolved to adapt to different populations of individuals. It is really a beautiful example of viral evolution. But okay, every year the flu virus evolves. It evolves based on this type of reassortment process. And the reason it does this really so differently than probably coronavirus is that it keeps circulating back into pigs and chickens and back into humans. So it's constantly moving back and forth in our livestock. And so this interaction between humans and livestock also creates more opportunities for flu to change. In addition, we have 4 different strains of flu that are also perpetually reshuffling. And so this is why we need to have a new vaccine every year. Now for COVID or SARS-CoV-2, the story is not quite as complicated. We have seen potentially some evidence of reassortment, but it's not really clear that it's happening in any way or shape or form like for flu. What we have here are individual little mutations that are occurring. These are little evolutions in the virus. Each of them look like simple adaptations to basically binding to the human ACE2 receptor better. Now here we do have evolution rates that do approximate influenza, but we don't see this reshuffling yet. And we only have one strain. So what that means is that it is possible that for SARS-CoV-2, unlike for flu, that eventually once we make a vaccine that is able to hit all the variants that can emerge, we might end up with just one vaccine in our lifetime, possibly administered in childhood, right? That will protect us for life. But it is also possible, and we have to keep tracking the rate of evolution of this virus, that we might for a few more years still need annual vaccines to just keep our immune system learning as fast as the virus can evolve to basically counteract this virus until we can get to this once in a lifetime vaccine. But we're working.
And some of the manufacturers of vaccines are already working on newer versions, and those do not, as I understand it, need to go through the entire regulatory process from the beginning, correct?
Right on, exactly. So we have multiple vaccine developers are working on these pan-coronavirus vaccines that will include variant spikes instead of the original spike, or even cocktails of different spikes that will end up giving us immunity against all the possible variants that we have across the globe. So that's going to lead to the induction of antibodies to lots of different targets. Okay, so let me just quickly go to the last slide, and then I want to leave time for questions, if that's okay. So I just want to just slide up, because I think this is really important. Now, I just want to make this point. We have been so ultra careful about safety during the development of these vaccines. We have included every single regulatory step to making sure that the vaccines that are being developed, even though they're going fast, that they really hit every milestone and every checklist that is required to make sure that they are safe. There is no evidence that these vaccines cause autoimmunity, zero. There is no data to suggest that the vaccines can alter any cells permanently. They are literally just software. No hardware changes ever occur. Vaccines do not interfere with other immune responses. Anaphylactic shock that we've heard from the Pfizer vaccine very early in the pandemic is super rare and only occurred in that one city in Alaska. And soreness, chills, muscle fatigue, all this other stuff is just proof that your immune system is working. So enjoy making antibodies. So that's where I'll end.
Yeah. Okay, that's great. Thank you very much. We do have a lot of questions I can see already being posted, and I had many more myself, but I want to be sure we get to the audience questions to give them a chance. There is one, though, that I'm going to ask you about, which is sort of about equity in vaccine distribution around the world. Many countries, including, the United States, obviously a lot of wealthier countries are ramping up their vaccinations. And some of the countries where we're working, vaccinations haven't even begun yet. So I wondered if you could address, the importance of equity in a global sense.
Yeah, so that's one of the hardest and most important questions I think that's on the on our radar. The issue right now has always been is that some of the vaccines that have come online the fastest require these really difficult, cold chain. They have these cold chain requirements that make them very difficult to distribute, right? And so the question is, what do we do in that scenario, and how do we basically make sure that we can get those vaccines out to the communities that, most likely require the vaccine really urgently, because these are populations of people that are as vulnerable or potentially more vulnerable because they don't have the luxury to social distance, nor do they have the hospital systems that can help to make sure that they're not going to, get sicker with a disease. So I think that where I think equity comes into play is the importance of, keeping the vaccine developers educated. It is our responsibility not just to educate, right, the globe about how vaccines work, but to keep educating the vaccine developers of how important it is to make sure that they understand who is vulnerable and where we can have the biggest impact. How can we, educate the WHO or keep them, kind of hold them to the fire to basically say that these are the populations that are, really that need the vaccine first. One thing that I have to say that I think is really exciting is the fact that these ad vectors are coming up online, right? The Sputnik vaccine, the vaccine coming out of China, as well as the J&J that's going to get approved hopefully this week, fingers crossed. I mean, those can get deployed so rapidly and
Much
so
more quickly, yeah.
all across the world. And so prioritizing that production, trying to work with governments to make sure that production timelines are biased towards producing the vaccines that are able to be deployed globally. I mean, that should be a priority. And so that is also where the media can help support the world and the global needs by trying to make that case to their governments to say, hey, these are vaccines that can actually be deployed. The other ones are really cool technologies,
Right.
but that is not what's going to be relevant for this particular global solution. And that's not to say that mRNA one day will not get there, right? Or that some of the protein-based vaccines are not going to be able to, evolve to be able to solve global problems, but we have to be realistic. And that is also, I think, our responsibility to make that case to the governments.
I just read a very kind of interesting story today about people in Britain which is of course one of the leading countries right now for vaccinations, who are either want the homegrown Oxford AstraZeneca because it's British, or they want what they're calling the posh vaccine, which is the mRNA vaccine.
Oh, gosh. That's cute. That's very cute.
So a question here that gets to something you and I talked about before the broadcast, which is about the Chinese Sinopharm vaccine. You did not mention it. Is there a reason for that, or why? What do you know about that one?
So the Sinopharm, that's the inactivated. Is that right, Patrick? Yeah. So
yeah.
Mm-hmm. Yeah. So sorry. So
it's Chinese, right? Yeah.
Yeah. So I did mention it in that original description of the 4 different kinds of platforms. Inactivated viruses was like the original design that Pasteur came up with. It was, this is really where we started off very early on with vaccine development. After Jenner came Pasteur, and that was the technology. The problem is with inactivated particles is that it's really hard to QC or to quality control to make sure that every single virus is inactivated. And so this is what's, led to a little bit of a movement from most big vaccine developers away from that technology. But that is one of the most, tried and true methods for making vaccines. And what makes that vaccine technology and platform so attractive, right, is the fact that you're giving the whole virus to the immune system to learn how to make immune responses against all the different components of the virus. Now, while we think that the spike is most likely the most important target for antibodies to try to fight infection, it is possible that there are other targets, especially on the surface of the virus. There's other proteins to which antibodies could bind and could drive immunity. So there might be advantages to the Sinopharm vaccine that we're not going to get with these other vaccines because you're getting this more holistic response. But, that has to be proven immunologically, right? That is currently kind of a tagline for the vaccine, but it is not immunologically proven that those other immune responses are beneficial. So I think what I would say to the media, what I would say to the community is I think that we should consider that part of the portfolio of vaccines. There is nothing to say that that vaccine is going to be worse. I just haven't seen the actual raw data from those trials to be able to say definitively that that platform is as good as data from the other trials that have been published in these rigorous journals. That being said, right, I think that we need to look at the data to really be educated and to really begin to understand what are the benefits of one platform over another, and how can we use these tools to build immunity in our populations as effectively as possible.
Okay, great. Another question about people who already have had COVID and how that figures into reaching herd immunity, the percentage of a population that maybe already have gotten it. And a follow-up question to that, should people who've already had the disease, whether it's severe or mild, should they get vaccinated as well?
Yeah, so the bottom line, at least from the recommendations here in the US from the Center of Disease Controls, is that even if you've had COVID-19 before, you have to get vaccinated and you should get both of the shots. Now, there is data that's emerging in the scientific community, in the medical community, suggesting that potentially if you've had COVID-19, you might only need one shot. But the problem is that natural immunity to COVID is really heterogeneous, right? It's so variable. Some people after COVID-19 have huge levels of antibodies. Some individuals have itty bitty levels of antibodies. And so because we cannot, evaluate every single person's level of antibodies before they come in to get their vaccine, it's really hard to say, oh yeah, you've had COVID-19, so you should only get one shot. What we want to do is we want to build immunity at the highest possible level in everybody, even if they've had COVID-19 before, because you want to make sure that immune response is impenetrable, right, to the virus to cause disease. Doesn't mean we're not going to get transmission, but we're going to try to block that disease. So I think until we can start to build in pre-antibody testing and we have an idea what level of antibodies is really critical, I would say that irrespective of what severity of disease you've had before, if you have the option to get a COVID vaccine, get it and do the full regimen. Don't hesitate.
And does the percentage of the population that has had the disease have an impact on herd immunity, on reaching herd immunity?
I mean, definitely, right? So the amount of immunity that's built up in a population will obviously factor in to how fast you can get to herd immunity. The problem is that we don't know how much natural immunity contributes to herd immunity because the responses can, in some cases, be quite transient, right? So what we think is that we should be relying on vaccine-induced immunity to get us to herd more readily than we should use natural immunity, because you're still advising everyone, even if they've had COVID-19, to go out and get vaccinated. So I think that it could factor in, but I would not rely on those numbers to get us there. I think we should really help the population understand the value of vaccination to get us to a point where we are, impenetrable again to that nasty virus.
Gotcha. Question from Hungary. Some experts there have said that inactivated vaccines that you talked about earlier could be dangerous for coronavirus because of the prolonged immune response and the cytokine storm. What is your opinion about that?
So this is, I think, potentially, it's a very important question, and I'm glad someone brought it up. And this is, again, is a lack of understanding of how these vaccines really work. So just like the protein-based vaccine, essentially what we do with these live viruses, they get in, they are not able to replicate, so they just go through one round. They basically go into the cell, they expose the immune system to the virus, and then it's cleared in the same way that proteins are cleared, protein vaccines are cleared. So it does not persist at all. The question about the cytokines is really interesting because it is a virus that's coming in. It does stimulate an immune response. And that stimulation is really what we're trying to mimic with those adjuvants that I mentioned in the protein-based vaccines. That stimulation of the immune system gets the immune system revved up. It gets it all ready to basically respond aggressively to these new proteins that are coming in. And it actually conditions them to make those proteins in a way that they can fight viruses much more effectively. So I would say to whoever says that those cytokines are potentially detrimental, I would say it's actually the opposite. Okay. And in fact, that those cytokines are likely to promote more effective immune responses against the virus because that's the native, or that's the natural response you would have to a virus. So the immune system is seeing this particle in the context of what a viral infection would look like, and it is going to condition to make the right flavors of immune responses that are going to fight the virus more effectively. So I think there are a lot of reasons to think that that type of response is actually beneficial. Okay. And there's no reason to think that it's going to persist and cause any kind of damage or problems.
Okay, great. Question about children. We're not vaccinating children yet. They haven't been approved for children. How important is vaccinating children to get us to herd immunity? And when will we be able to do that?
Okay, this is a brilliant question. Whoever asked, you're amazing. So we are never going to get to herd immunity without getting kids vaccinated. Bottom line, right? They are a massive population within all the different communities across the globe. If we don't get into kids, we will never get herd immunity. And what we've known and what we've learned over the last year since the virus has popped on the radar is that kids are carriers and they have very high levels of viral replication. So we need to get the kids vaccinated as soon as possible. There are pediatric trials that are starting across the globe. We are already working our way into the teenager population, right? Founded on lots and lots of data from HPV vaccination, so the human papillomavirus vaccine that has been unbelievably effective and super safe in teenagers. We are working our way with the COVID-19 vaccines down into that age group. And soon enough, I think we're going to be into the smaller kids and even potentially into infants in the near future. And we are working our way into the pregnant women populations, which are obviously highly vulnerable because they develop severe disease. So there is zero question that if we don't get it into the pediatric population, we're never going to get to herd. And so that is a top priority from global communities and vaccine developers.
Great. Last question, because I know you have a hard stop. There have been some countries or municipalities, states in some cases, are making the decision to try to get as many first doses into as many people as they can before giving second doses. What is your opinion about that? Should we be trying to make sure everybody gets 2 doses or just get it into as many arms as we possibly can?
Oh man, that's a really tough question because I think there's so many, just so many opinions on this topic. So, okay, I don't want to bias anyone. So I think that the way that I can put it this way is I think there's a lot of epidemiological modeling going on, and I just believe the data. The data is the data, and that's what we should be following. And there are 2 camps in the epidemiology space. There's the one camp that believes that by standing up immunity in as much population as possible, we're going to get a little bit of resistance against the virus. And that's going to give the population a little bit of a bump and potential ability to try to reduce some of the severity of disease. The other camp in the epidemiology space says, let's hit the most vulnerable populations that are most likely to get really sick and potentially die. And by standing up complete immunity in those individuals, we'll be able to keep people out of hospitals and off their deathbeds. And I think that where we are right now is we're seeing, unfortunately or fortunately, a global experiment happening where different communities are testing each of these hypotheses. I don't know which one is going to work best, but I think it has to be based on the ethics, morals, and principles of that population that has to decide which one they feel more comfortable with, because neither is perfect. Right? Giving the doses to only a small fraction of the people that we think are the most vulnerable does not protect those other individuals that might not get vaccinated from potentially having that unfortunate outcome. And only giving low-level immunity to the entire population is not necessarily going to protect people who are most vulnerable either. So neither is perfect, but you got to make a decision because we cannot hold vaccine doses back. And so I think it's a population, it's a personal choice. It has to be made by that community.
And we'll get more evidence on it too, as we find out whether that 1 dose only is going to work. Okay. Well, unfortunately, we do have to stop. We could go on for hours on this, and this has been so educational, Dr. Alter. I really, really appreciate your taking the time when I know how busy you must be right now. So thank you so much for being with us. Really, I've got a ton more questions. I can't even count them all that we were not able to get to. So obviously a lot of interest in this topic, but you've really educated us to a great extent. Feel free to leave. I know you have a call. I'm just going to do a little bit of wrap up stuff, but we really appreciate your.
Bye, guys.
Okay. I just want to remind everyone that this webinar is recorded and it will be found on ICFJ's YouTube channel within 24 hours. We also want to remind you to fill out an audience feedback survey that we're going to send all of you who registered for this webinar, because your feedback is really important to us as we line up future webinars. We want to know what works for you and what doesn't. And I want to again thank Dr. Alter. I want to thank all of you for your questions. Like I said, as you can see, those of you who are following, there's so many questions in the chat that we weren't able to get to, but I hope this presentation has been really useful to you. Please stay tuned. We'll continue doing webinars, both this kind of webinar that address scientific and medical questions. We're also doing a lot of training-oriented webinars for those of you who want to learn new skills in how to tell better stories, how to use data, cybersecurity, all kinds of issues that we'll be offering webinars on the training side. So thank you all for joining us, and we look forward to seeing you again very soon, and please stay safe.